species that currently are described, or even more species. And as you probably know, it's a book example, lizards represent a book example for really high rates of speciation and adaptation. So you can find these very diverse communities where you can have up to 15 species that are coexisting. And despite these communities where you have several closely related species, hybridization in Anolis is very, very rare. Currently, we know only about 8 species pairs that hybridize, which is a very small portion considering that we have 400 species. It is believed that the very low degrees of hybridization are caused by very effective pre-mating isolation systems. In particular, so in here you can see a male anole, and it's using its colorful dewlap and head bobbing to impress the female. And the female is capable of recognizing its own species, and that reduces or even eliminates mismatings and therefore hybridization. And these effective pre-mating isolation systems are also believed to promote the high speciation rates in our lizards. So today I'm going to talk about 2 species of anoles from Puerto Rico. These are the 2 species. It's Anolis pulchalus and Anolis krugai. If you are familiar with the different ecomorphs of Anolis, these represent one of the smallest species. They are called grass bush anoles. And they are sister taxa. So in here on the tree, you can see that they diverged 5 to 10 million years ago. In here you can see in this map that they are sympatric. So these dots represent occurrence localities, but also our sampling localities. And you can see that they are highly sympatric, and I can also tell you that there are many times they are also syntopic. They have very different dewlaps, and it also explains my coloration, my choice of coloration. So from now on, throughout the talk, anything that represents Anolis pulchalus will be red, and anything that represents Anolis krugai will be yellow. And given that these big differences in the dewlap and what I have told you about hybridization and female choice in Anolis, it was very surprising to us to find that these 2 species hybridized or have hybridized. In particular, we found these individuals that look like just like pulchalus, so you wonder how we know they hybridize, but they have a krugai, they have krugai mitochondrial DNA. And the only way how to achieve this is that they must have, at some point, they must have hybridized with each other. In particular, we have male pulchalus hybridizing with female krugai. And the pattern of this mitochondrial introgression is actually very interesting. So first of all, we found out that it's pretty extensive. So we have possibly 1/3 of the species range are these introgress individuals. I will call them hybrids. So 101 individuals, that's 1/3 of our sampling. And what is even more interesting is that we have this complete replacement of the native mitochondrial DNA with the foreign krugai DNA throughout many, many populations, in particular 14. So we identified 14 populations with complete replacement, 2 populations on the edge of the distribution that show a mixture. So of course our question was, when did this hybridization happen? So we performed an AMA analysis to look at the divergence between the mitochondrial plates of Anolis pulchalus that have the foreign mitochondrial DNA and Anolis krugai, and we found out that the hybridization happened pretty recently, at least for us, because these species diverged some 5 to 10 million years ago. So in here you can see that the peak is around So we estimated the number of events, and we did it very, very conservatively and came up with a number of 15. So our very conservative minimum number of hybridization events is 15. In reality, we are probably talking about hundreds, maybe thousands of hybridization events. So these guys hybridize and they stay in place. They have not been moving around much. So of course, our next question was, what is the mechanism? And when you talk about mitochondrial DNA introgression, the first thing that comes to your mind is genetic drift, which is usually driving mitochondrial introgression. So in here we have a hypothetical scenario where we have a species, so Pulcellus would be in red, and we have a hybridization that is probably not going to be very common, so we will assume it's about 5%. That seems like a reasonable number of frequency of hybridization. So now my question is, what is the probability of fixation of the foreign mitochondrial DNA? Population genetic class. So you probably remember this. It's probability of fixation equals the alias origin of frequency, right? Population effective size doesn't necessarily play a role here. So in this case, it's going to be, in each population, it's going to be 5%. Well, if we have a lot of different populations, then we have to multiply it by number of populations. So it's going to be 0.05. I'm aware that this is a very rough estimate. We don't really know what the original frequency was. Times 10 to 14, which is number of populations where we have the fixation. So my point here is that to have a fixation in so many populations at the same time is very, very unlikely. It's basically zero. So based on these patterns, we came to the conclusion that genetic drift is just not responsible for the mitochondrial introgression in these two anoles, and that there must be some sort of selection. So what mechanism or what type of selection can be going on? So I apologize that it's not very well visible, but the first possibility is that we have a negative selection against deleterious mutations on the mitochondrial DNA. We can have positive selection on traits that are affected by mitochondrial genome, and we can also have a positive selection on the joint mitochondrial nuclear genome. And in here we kind of hoped for the third one, so we decided to look at the genomic composition of these hybrids. In here is a warning, I'm not going to solve this problem. This is where we are regarding the mitochondrial DNA. We don't know what's the nature of the selection, but the analysis of the nuclear genome actually brought some even more peculiar properties of this hybridization. So let me repeat what we have learned so far. So we have these guys that look like Pulcellus, but they have foreign mitochondrial DNA. So we were aware from the beginning that probably the genome is going to be Pulcellus prevalent. That the most markers are going to be Pulcellus-like. So we came up with two different hypotheses. The first one is backcrossing. So we predicted that if backcrossing is going on, then the F1 hybrids are going to be a mixture of both parental species. They're going to backcross with male Pulcellus. This is a little problematic because we already know that the male Pulcellus are not present. They were outcompeted, right? We have only the hybrids within the range. But we can assume that at least at the beginning, before they were gone, that they could have backcrossed some. And then after n generations, we will have something like that. We will have mainly Pulcellus genome, but we'll have some Pulcellus markers. They are going to differ in different individuals based on how many backcrossings and stochasticity. Our second hypothesis was limited introgression. So there are cases where when two parents hybridize, then the proportion of the genomes is not equal. We can have one genome severely underrepresented. Studies suggested it's about 5 to 10%, and the causes are chromosomal divergence or negative interactions among loci and so on. So that was our second hypothesis, that we will find underrepresentation of one of the genomes, and similar loci will be introgressed across independent hybridization events. So in here, just a repetition. So this is our mitochondrial tree. Notice that the hybrids here nest with anolis krugai. So in here I color-coded them light blue because I hope it would be better visible. It seems like it would if I look at the nuclear tree, so it was based on about 10,000 SNPs. It's the exact opposite. So you can see that the hybrids are nesting with pulcellus, same as what you would expect based on the phenotype, and krugi are completely separate. So where are the krugi loci? So we put a lot of faith in our fast structure analysis, which is another version of structure. You already saw quite a bit of that, so you know that I'm going to show you genomic composition of krugi, hybrids, and pulcellus. And you saw that the hybrids usually exhibit a mixture of both parents. Now I'm going to show you probably the most boring structure analysis in the history of structure analysis. It's nothing. So we found out two groups, krugi, pulcellus. The analysis couldn't even believe what I'm asking for because the hybrids are basically 100% pulcellus. We did this on two different data sets. We even tried to analyze chromosomes separately. It was always like that. Other analysis of this is a principal coordinate analysis, very similar results. Hybrids are clumping with pulcellus, krugi completely separate. So in here I could show you analysis by analysis, which I'm not going to do because they all resulted in exactly the same. So we used two data sets, one medium number of SNPs, one a lot of SNPs. We used a lot of different analysis, and we completely failed. We didn't find any krugi loci. So at this point, we realized that the results are just not consistent with our two predictions, and that we have to come up with a third one. And we did. It's called genome exclusion hybridization, where one of the parental genomes is completely excluded. This type of hybridization has not been yet reported in any vertebrates, which makes it even more exciting. It has been reported in frogs and in some insects. But for us, what is very important is that it might work as an effective reproductive isolation mechanism. So if there is no genetic introgression, that's as if the hybridization never even occurred. So in here I'm going to finish with our future work in a form of a question. So as I told you at the beginning, we believe that premating isolation, the female choice, is the one that promotes high speciation rate in anoles. But if we have a type of a hybridization that's actually completely excluding one of the parental genomes and working as an effective reproductive isolation mechanism, is it possible that genome